And he's now using that to select drugs for those patients. And so, whereas the organoids were not affected because they died when you give them direct exposure of the same dose of drug that a patient would see. So that's another use. It's not just to replace animal testing. And then finally, I think the game changer in drug development is right now, big pharma will spend hundreds of millions to develop a drug, tens of millions on a big clinical trial, and they almost always fail. And what they do is they have statisticians number crunch the results and say, was there a genetic subpopulation or a group with similar phenotype that responded better than the others? And if they find it, they'll do a narrow trial, and if it works, they get approved for a narrow application. With patient-derived cells and organ chips, you can imagine from the beginning, start out with a low number. Let's start out with 100 people, men a certain age who have a certain type of IBD, inflammatory bowel disease, and you have drugs that you're developing tested on them. Well, maybe 50 respond really well, the other 50 don't. And then take their liver chips and kidney chips to look for toxicities and find 35 who have no toxicity and respond well, and use them for a clinical trial.